Hello and welcome to this module on classic rapid amplification of cDNA ends, or RACE for short. So let me begin by discussing an overview of RACE. So RACE in concept is pretty simple. The idea is that you generate complementary DNA from messenger RNAs using reverse transcription, and then you amplify the cDNA product via PCR or polymerase chain reaction. You can then perform analysis of your amplified products via Southern blotting. So let me begin by discussing the classic 3' RACE. As I said earlier, the first step in RACE is reverse transcription. So in the case of 3' RACE, reverse transcription is indeed the very first step, and that's where we create the complementary DNA to our messenger RNA of interest. Now, to perform the reverse transcription, we use what is called a hybrid primer, which is also called the QT, where the T stands for total, and I'll let you know why it's called total in a bit. Now, the hybrid primer, or QT, or Q-total, consists of a sequence that has a part that consists of an oligodeoxythymidine. Here you can see that the hybrid primer consists of 17 oligodeoxythymidine bases, as well as a unique oligonucleotide sequence, which consists of an internal part called QI and an outer part called QO. Here you can see the actual primer, the QT. QT consists of all of this. It has an oligodeoxythymidine, or in other words, a bunch of thymidine bases. You have a Q internal sequence, that is, it's internal, it's not towards the 5' end, and it has also a Q outer sequence, which is also called the QO. Now, importantly, the unique oligonucleotide sequence, that is the QI and the QO, is termed the adapter or anchor primer in various different research papers. So depending on the research paper and the author, they may call it an adapter primer or an anchor primer. Now, importantly, this together here is a single primer. So in the case of QT, this is a hybrid primer that has different parts, but within the unique oligonucleotide sequence are the Q internal and the QO, and both the Q internal and the QO will be used separately as primers in the amplification steps of our cDNA product. So the idea with the classic 3' RACE, as is with 5' RACE, is that you start with a messenger RNA, as you can see here. Now, when you have the messenger RNA, you want to produce a complementary DNA out of that messenger RNA, and to do so, you perform reverse transcription using a reverse transcriptase and the hybrid primer that I highlighted before. So the product will be our first cDNA strand, and the first cDNA strand obviously will have the hybrid primer embedded into it, and of course the cDNA strand. Importantly, you should recognize that this first strand of cDNA is negative sense. Recall that a negative sense strand is reverse complementary to the positive sense strand and the RNA transcript. So this is something to keep in mind. Of course, the exception that you have to keep in mind here between the negative sense strand and the RNA transcript, or the messenger RNA, is that in the cDNA, you will have thymidines, whereas in the messenger RNA, you will have uracils. But other than that, these are completely reverse complementary to one another. In the case of a positive sense strand and its relation to messenger RNA, which we don't have a positive sense strand here yet, the positive sense strand is the one that looks exactly like the messenger RNA transcript, again, with the exception of uracil and thymine differences between the messenger RNA and the positive sense strand. And one more side note, just keep in mind that the positive sense strand, while it looks like the messenger RNA transcript, does not code for the messenger RNA and for the subsequent protein translation. It is actually the negative sense strand or the antisense strand that is used as a template for messenger RNA and subsequent protein translation. And this should make sense considering the fact that I just told you that this strand is a negative sense strand. And we have performed reverse transcription, meaning we did the opposite of transcription and we went back to the very identical DNA sequence that produced this messenger RNA. But anyways, that is not the topic of this discussion. So I'll move on now to the amplification step of cDNA. Now, the amplification of cDNA in classic 3' race is performed via two sets of amplifications. So in the first set of amplification, we will use two primers. A primer that is specific to the gene of interest, and we'll call this the GSP1, as well as part of the adapter primer. Recall the adapter primer is the Q1 and the QO together, and that part of the adapter primer that we will use in the first set of amplification is the Q outer or the QO. So it can be a little bit confusing to try to understand what's happening using this diagram, as it does not represent the typical diagram that we see in the case of PCR, primer annealing, and extension, simply because this primer, for example, doesn't look like it is annealing to any sequence. It just simply seems to be extending a sequence by itself. And then you have the QO primer that also seems to be extending a sequence by itself. But in reality, what the GSP1 primer here will anneal to and bind is a specific region in the negative sense strand of the cDNA, which is precisely this region. And it will extend this way such that it produces a positive sense strand of the complementary DNA. On the other hand, you can see here that you have a QO. Now, the QO here is binding to the 3' end, and it's extending this way. But if you think about it, this QO cannot possibly bind or complementarily bind to this QO sequence. So what this QO is actually binding to is the reverse complementary sequence of the QO that we see in the hybrid primer. So in here, once the GSP1 has extended in this region, you will have, for example, here, you will have A, and then you will have the reverse complementary sequence of the QI. So you'll have the reverse complementary sequence, and then you'll have the reverse complementary sequence of the QO. And since you have the reverse complementary sequence of the QO, this QO can actually anneal to its reverse complement and extend this way. This is the basics of the first set of amplification in classic 3' race. In the second set of amplification of the cDNA, you will use another part of the adapter primer, which is the Q internal, and you will also use another primer that is specific to the gene of interest, which is the GSP2. Importantly here, what you should recognize is that the idea of the second set of amplification is that we use what's called nested primers. And by nested primers, we mean that these are primers that are going to be more internal to our amplified cDNA products, such that the amplification minimizes non-specific amplified products. And so our amplification of the cDNA is going to get more specific, more internal to the parts of the cDNA relating to the messenger RNA that we care about. So in the second set of amplification, you will have a Q internal part, and you will have the GSP2 primers. These are both primers here. So the GSP2 primer, well, actually, let's begin with the Q internal part here. It's important to recognize that the Q internal part cannot possibly anneal to this strand, i.e. the negative cDNA that we have here, because in this section right here, you have another Q internal, and a Q internal sequence is not going to complementary bind to an exact identical sequence to itself. So what the Q internal here is binding to is the positive sense strand. So it is annealing to a positive sense strand. And so it's annealing to a positive sense strand that's going to be created by the GSP2. So the GSP2, as you can see here, is going to target this specific region, and it's going to anneal and extend this way. And then here you will have a reverse complementary region of the Q internal, or I should say, a reverse complementary sequence of the Q internal, where the Q internal can bind and extend. So I hope this makes this particular part of the PCR diagram more accessible. And now I'll move on to the classic 5' race. This concludes the classic 3' race. We've done reverse transcription, followed by a first set of amplification, followed by a second set of amplification with nested primers that are more internal. So the classic 5' race is similar, albeit it has more steps and maybe a little bit more complicated. In classic 5' race, as we did in classic 3' race, we started with reverse transcription, which is the process of cDNA creation. And to perform the reverse transcription, we're actually going to use a gene-specific primer. Recall that in classic 3' race, we use the hybrid primer with the oligodeoxythymidine or the oligo-DT, followed or connected to the adapter primer. However, in this case, we are using a gene-specific primer. So in this case, here you can see the gene-specific primer. It's annealing to this portion of our messenger RNA of interest, and it's going to extend this way to produce our very first strand of cDNA. Something that I may have forgot to mention in the previous discussion of the 3' race is that once you have the reverse transcription performed, you will use RNase in order to degrade the messenger RNA, such that you only have the cDNA, or the negative sense complementary DNA. So now we have our complementary DNA. This is a negative sense strand, as I've discussed before. Why that is? This is a negative sense strand since it's the reverse complementary sequence of the messenger RNA. And the messenger RNA is encoded by the antisense, or the negative sense strand, in a double-stranded DNA genome. And as you can see here, you have the gene-specific primer called GSP-RT, RT for reverse transcription, included within our first strand of complementary DNA. Now, once we create the first strand of cDNA, we will then add a poly A tail to the 3' end using terminal transferase. So it's important to recognize that terminal transferase is more or less an enzyme that adds nucleotides to the 3' end of DNA or RNA molecules in a template-independent fashion. So in other words, it can add nucleotides without requiring a specific template sequence. That's why you don't see any template here. You see that the poly A tail has been added without any template, and it's been added to the 3' end of the cDNA. Once we've added the poly A tail, we can then perform the amplification of the cDNA, and similar to the 3' race, we will perform a first set of the amplification of cDNA, followed by a second set of amplification of cDNA using nested primers. So let me begin by discussing the amplification of cDNA in the first set. It can be confusing here as we're using 3 different primers to amplify cDNA, which is something that might be unheard of when we're considering classic PCR of a double-stranded DNA, where we simply use 2 primers and extend. So in the first set, we will use a hybrid primer, QT, which is the same concept as we saw in the 3' race. We will also use a QO primer, which represents a sequence that is identical to the QO part in the hybrid primer. And we will also use a gene-specific primer, which we'll call GSP1. So here is where it might be useful to just use some reasoning to understand what's going on here. So the first thing to recognize is that the first thing that's going to happen here in terms of the cDNA that has a poly A tail added is that it's going to anneal with our hybrid primer here to form a second strand of cDNA. So in this case, this is our negative sense strand. And what's going to happen is that the hybrid primer is going to anneal, specifically it's the oligodeoxythymidine that's going to anneal to the poly A tail and then extend this way to form a positive sense strand. Now, once that happens, you can have the GSP1 anneal to this positive sense strand in this region. As you can see that the GSP1 is getting more internal. So now we don't have, for example, the GSPRT or the gene-specific primer used in reverse transcription. It's no longer in our cDNA amplified product. And so this GSP1 will extend this way, creating a negative sense strand of our cDNA. Now, you might be wondering, well, when does the QO play a role? Well, obviously, the QO here will not anneal, for example, to anything in here because we don't have any sequence here for the QO to anneal to. In addition, it cannot possibly anneal to the QO here within our hybrid primer because QO cannot complementary base pair with QO, which is an exact identical sequence. So the only explanation here is that the QO will anneal actually to the negative sense strand created by GSP1. In other words, it will anneal to the negative sense strand created by the GSP1, where you have a reverse complementary region of QO where QO can bind to. So this would be the region right here where QO binds to. This is a region where you have a reverse complementary sequence of QO, so it makes it so that QO can bind to it. Once we're done with the first set of the amplification, we can move on to the second set of the amplification, where we're going to use more internal primers. Here we'll use the Q internal primer and the gene-specific primer 2 or GSP2. As you can see here, you have the Q internal that is annealing to one of our cDNAs and extending this way. By simple reasoning, we can deduce that this Q internal primer must be binding to the negative sense strand, the bottom strand here, to the Q to the reverse complementary Q internal region. So here you have a reverse complementary Q internal region, and this will anneal with our Q internal here, and the Q internal will extend to the right. On the other hand, you have the GSP2, and the GSP2 can anneal to the sequence created by extended by the Q internal primer. So it can anneal here and it can extend to the left, producing our cDNA. Obviously, the top strand here would be a positive sense strand, and the bottom strand here would be a negative sense strand. This concludes my module on classic 3' race and 5' race.